So my presentation is on the neurobiological basis of emotion recognition and its modulation by contextual visual information. Emotion recognition is a very interesting topic. The problem is that traditionally many studies have relied on simplistic stimuli rather than more naturalistic, dynamic, animated stimuli like changing facial expressions of people and or potentially incongruent contextual cues. So we decided to investigate this using multiband fMRI with 29 healthy human subjects. So for a single trial, a 2 seconds video of a facial expression was presented, which could be either happy or neutral or sad. This was to test for emotion recognition. This was preceded by a static image, a scene of people with blurred out faces, as shown here, which is the contextual cue, the contextual visual information. Again, this could be either happy, neutral or sad, and in some conditions there were no cues present. It was just the face video being presented. And so subjects had to press the correct button indicating the emotion of the face video within the face video time window. So with regards to the behavioral data results, as shown here in figure 3, what was found was that participants responded faster to happy face videos compared to neutral, and slower with sad faces. The other main result was for when comparing congruent and incongruent conditions. Congruent conditions were conditions when both the face video and the context cue were emotionally congruent, for example happy and happy, or if they were both neutral or both sad. Incongruent conditions would be if they were different, for example a sad image cue and a happy face video, as shown here. So you can see incongruent conditions were slower, but this difference was not statistically significant, surprisingly. So now for the neural data, it is shown here. So firstly, just to clarify that what was modeled was the bold signal change between the onset of the face video up until the subject pressed the button indicating their response. So essentially what was modeled was the response time duration. Okay, so for the effect of emotional salience in faces, happy and sad combined, and regardless of whether the prior context was congruent or incongruent or absent, there was no significant activity with regards to the regions of interest laid out in hypothesis 3. However, with whole brain exploratory analyses, there were some significant clusters. So in figure 4, you can see that behavioral performance or response time was positively associated with neural responses in the bilateral occipital-lingual area, past the fusiform gyrus, as well as supplementary motor and cingulate cortical areas. Looking at figure 5, you can see that happy face videos implicated similar regions, but also the superior, middle, and inferior frontal gyri, inferior parietal supramarginal cortex, and a region overlapping with the anterior insula. So emotional faces recruit distributed clusters in different brain networks. Networks involved in affective or emotive processing, networks for sensory visual processing, and ones for premotor related, action planning related processes. It's very important to understand here that we are interested in not just brain regions involved in emotional conditioning or empathy or emoting, but we are interested in the evidence gathering and decision making process that underlies the active recognition of distinct visual features, especially with socially relevant dynamic stimuli such as facial expressions. And for neutral faces, see figure 6, there was also task-related activation in a separate circuitry, primarily involving the superior parietal and middle frontal areas. And then for incongruence, in figures 7 and 8, there was an association with or recruitment of the middle and anterior cingulate cortex, the superior and middle frontal cortex, the orbital frontal cortex, and parietal and occipital regions. And finally, figure 9 just shows occipital-temporal activity for face videos which were not cued of context. Okay, so to conclude, at the psychobehavioral level and at the neuroanatomical, neurophysiological level, the emotion recognition process varies based on different facial emotions. In other words, it is valence dependent. And it is also differentially affected by varying types of contextual visual information. So with regards to facial emotions, our behavioral data are consistent with other works in electrophysiology and eye gaze tracking, which showed how happy smiling faces may be recognized more quickly based on the way facial features are sampled. With the neural data, these systems here, for processing dynamic emotional stimuli, as I explained earlier, also do partially overlap with the literature. Interestingly, for neutral facial expressions, some studies actually show that compared to neutral static non-face objects, neutral faces may have their own emotional salience properties, and compared to emotional faces, they may recruit their own distinct parietal and frontal brain regions. For the effect of incongruent cues with the neurobiological data, there was some overlap with the scientific literature. However, the weak behavioral effect is presumably because the different contextual cues simply did not have that much effect on the face video response. This may be due to the cue's emotional valence being ambiguous because of the blurred faces, or subjects not really attending the cue that much, or it's most likely because of how incongruence and context is defined in the emotion recognition literature. So, for example, some studies define it as human body language or scenes or voices presented at the same time as facial expressions rather than before. So that may be why we obtain different results. So one of the limitations is that we were not able to extract the original data for some of the runs for 6 subjects out of the 29, which were locked in the MRI scanner system, and we could not get access to these due to the COVID-19 lockdown, so we did lose some statistical power here. Although according to previous studies, 23 subjects had been shown to be a large enough sample size. On the other hand, these 6 subjects that were lost actually had relatively high behavioral response accuracy relative to other subjects, which means that those may have provided more accurate data. But this makes the assumption that better behavioral data somehow correlates with better neural data, and further investigations would be required to investigate that further. Another limitation is not accounting for between subjects and within subjects variability with certain confounding variables, such as the subject's attention level or task expectations. Another issue is the automated anatomical labeling used, or even the large susceptibility artifact, the air tissue signal dropout in multiband fMRI that affected the inferior medial PFC and the OFC, regions that we were particularly interested in. There's also other general limitations relating to paradigm design and procedures, fMRI data being only an indirect measure of neural activity and only correlational rather than causal, and so on. So one thing that could be done is getting back the data for those 6 subjects that were not extracted. We could also use a different type of processing pipeline, including other types of analyses or modeling, because some studies suggest that different research labs can come up with vastly different final results, even with the same raw data and hypotheses, which is a potential problem when replicating cognitive neuroimaging studies. As for pursuing further experiments, we could use stronger cognitive paradigms for the contextual information. We could investigate other emotional faces like angry and fearful ones, more trials could be performed, and so on. And we could also combine fMRI with electrophysiology so they can complement each other's spatial and temporal resolution limitations, and for greater ecological validity as well. Or even use MRI-based connectivity analyses, for example with diffusion tensor tractography or resting state fMRI data, especially when looking at altered connectivity in clinical populations, for example for prosopagnosia, alexithymia, autism, social-emotional disorders, etc., and perhaps even use pharmacological approaches as well. And finally, we could use deep machine learning and artificial intelligence, on the one hand to process the fMRI data, and on the other hand to produce computational models of biological neural networks and simulate the emotional recognition algorithms of the brain.